Development of brain structures and anatomical basis of neural tube defects. So in this we discuss the development of brain structures. So the competency is development of neural tube, spinal cord, medulla oblongata, pons, midbrain, then cerebral hemisphere as well as cerebellum. The first indication of neural plate in human embryos is appearance of median sulcus, around 23 days of development. Rostral half represents the forebrain and the caudal half hindbrain. Closure of neural groove begins near the junction between the brain and spinal cord. Rostrally and caudally, the cavity of developing neural tube communicates via the rostral and caudal neuropores with the amniotic cavity. Then closure of neuropores. Rostral neuropore closes at about 30 days. Caudal neuropore closes at about 1 day later. The site of final closure of rostral neuropore is at the site of the embryonic lamina terminalis. Closure of the neural tube in human embryos proceeds both rostrally and caudally. Neural tube layer formation. Initially, the wall of neural tube consists of a single layer of neuroepithelial cells or matrix layer. Mitosis occurs on the internal side of the cell layer only. Migrating cells from the second layer form a second layer around the original neural tube. The new layer is the mantle layer or intermediate zone. It becomes progressively thicker as more cells are added to it from the germinal neuroepithelium that is now called as ventricular zone. So the cells of the intermediate zone differentiate into neurons and glial cells. These neurons send axons into outer layer, the marginal zone. Mantle layer containing the cell bodies becomes the grey matter. Axonal marginal layer forms the white matter. In the spinal cord, this three-zone pattern is retained. Then, development of the spinal cord. After neurulation, the spinal cord can be divided into dorsal RR plates derived from the lateral parts of the neural plate and the ventral basal plates derived from the medial parts. The alar or basal plates are separated by sulcus limitans of His. Alar plates and incoming dorsal roots form the afferent or sensory part of the spinal cord. Basal plate and its exiting ventral root form the efferent or motor part. Early spinal cord reaches the end of the vertebral column. During fetal period, it ascends to sacral and later lumbar levels. Then early development of the brain. Forebrain divides into telencephalon and diencephalon that can be identified because it gives rise to optic vesicles. Cerebellum. The development of cerebellum takes place largely in the fetal period. The cerebellum arises bilaterally from alar layers of the first rhombomer. During third month of development, growth of midline component accelerates, thereby forming the vermis. By approximately 44 days, the internal cerebellar swellings contain dentate nucleus. Superior cerebellar peduncles can be seen around 48 days. Cerebral cortex. Each hemisphere first grows caudalwards, then bends to grow in the ventral and rostral directions. In this way, the temporal lobes arise. The outgrowth of the caudate nucleus, the amygdala, the hippocampus, and the lateral ventricle occurs in a similar C-shaped way. During the fetal period, the complex pattern of sulci and gyri arises. On the lateral surface of the brain, the sulcus lateralis and sulcus centralis can be recognized from 4 months onwards. On the medial surface, first the parieto-occipital and the cingulate sulci appear, followed by calcarean and central sulci. The formation of sulci and gyri in the right hemisphere usually precedes that in the left one. Usually, the pallium is divided into medial pallium or archipallium, dorsal or neopallium, lateral or paleopallium. Recently, an additional ventral pallium was added. Medial pallium forms the hippocampal cortex. Dorsal pallium forms the six-layered cortex, cerebral cortex. Lateral pallium forms the olfactory cortex, and the ventral pallium forms the claustro and amygdalar complex. Corpus striatum. Subpallium consists of two progenitor domains, the lateral and medial ganglionic eminences generating striatum and pallidum respectively. The hippocampal formation comprises the dentate gyrus, the hippocampus, the subiculum and parahippocampal gyrus. These structures develop from the medial pallium and are originally adjacent to cortical areas. Hippocampal formation and phonics. During the outgrowth of cerebral hemispheres, first caudal verts, subsequently ventral verts and rostral verts, the retrocommissal part of the hippocampal formation becomes situated in the temporal lobe. The rudiment of the supracommissal part of the hippocampus can be found on the medial side of hemisphere on the top of corpus callosum, that is the indusium griseum. Thank you.